Hello, everyone. Today, I am pleased to present a study entitled Single-Cell Profiling of Peripheral Blood Reveals Acute-Phase Immune Dysregulation in Human Scrub Typhus. Scrub typhus is a potentially life-threatening, mite-borne disease caused by Orientiatsutsugamushi, a gram-negative obligate intracellular bacterium. After being inoculated into the skin during a mite bite, the bacterium infects local host cells, often causing a characteristic eschar, and may subsequently disseminate throughout the body. Although scrub typhus has traditionally been associated with the Asia-Pacific Tsutsugamushi Triangle, growing evidence suggests that its geographic distribution is broader than previously recognized. However, the cellular and regulatory mechanisms shaping the host immune response remain poorly understood at single-cell resolution. To define acute-phase immune dysregulation in scrub typhus, we integrated single-cell RNA sequencing, spectral flow cytometry, TCR/BCR repertoire profiling, and plasma proteomics using peripheral blood samples from patients and healthy controls. This multiomics approach allowed us to examine coordinated changes in CD8 T cells, CD4 T cells, B cells, and myeloid cells, and to identify immune cell populations associated with bacterial burden. In this study, peripheral blood mononuclear cells from patients with acute scrub typhus and healthy controls were analyzed using single-cell RNA sequencing and immune-receptor repertoire profiling. Spectral flow cytometry and plasma proteomics were further employed for independent validation at the protein level. After stringent quality control, more than 61,000 cells were retained for subsequent analyses of immune cell composition, transcriptional states, and disease-associated molecular alterations. First, we observed extensive remodeling of the peripheral immune landscape. Patients showed a significant contraction of the B-cell compartment and a relative expansion of myeloid cells. Although the overall T-cell proportion remained relatively stable, the distribution of T-cell subsets changed markedly. Cell chat analysis also predicted an increased number of ligand-receptor interactions but reduced aggregate signaling strength in myeloid cells, B cells, and CD4+ T cells. These findings suggest extensive but potentially dysregulated immune communication during acute infection. The most prominent adaptive immune feature was disruption of the CD4 to CD8 T cell balance. Patients showed a reduction in CD4+ T cells and expansion of effector memory and proliferative CD8+ T cell subsets. These expanded CD8 T cells expressed high levels of cytotoxic molecules, including GZMA, GZMB, PRF1, and granulysin, together with activation markers such as CD38 and TNF-alpha. At the same time, exhaustion-associated molecules, including LAG3, CTLA4, and PDCD1, were also increased. T-cell receptor analysis revealed oligoclonal expansion and reduced repertoire diversity. Larger CD8 T-cell clones showed stronger activation, cytotoxicity, and exhaustion-associated signatures. Together, these results indicate the expansion of a highly cytotoxic and clonally amplified CD8 T-cell population with exhaustion-associated features. The B-cell compartment showed a different pattern. Acute infection was associated with depletion of naive B cells, increased apoptosis and FOS expression, and expansion of plasmablast and plasma cell populations. We also identified pronounced inflammatory remodeling of the myeloid compartment. Patients' overall myeloid proportion remained relatively stable. Different myeloid subsets displayed inflammatory, antimicrobial, antigen presentation, and cell death-related programs. Flow cytometry further demonstrated increased expression of CXCR6, CCR7, granulysin, and IL-1 beta in CD68 positive myeloid cells. Importantly, the frequencies of both CD14+ CXCR6 and CD14+ granulysin positive myeloid cells were positively correlated with bacterial burden. These populations may therefore represent candidate indicators of infection-associated myeloid activation. In summary, our multiomics analysis reveals coordinated remodeling of both innate and adaptive immunity during acute scrub typhus. The key features include the expansion of cytotoxic and clonally amplified CD8 T cells with exhaustion-associated characteristics, depletion and apoptosis of CD4 T cells, B-cell contraction with terminal differentiation, and inflammatory myeloid remodeling. These findings provide a framework for understanding peripheral immune dysregulation in scrub typhus and help prioritize candidate cellular markers and immune pathways for future longitudinal and functional studies. Thank you very much for your attention.